The complement system, it is a major part of our innate immune system. The research on this complement began in the 1890s, and the major contributions were made by two scientists, Paul Ehrlich and Jules Bordet. They independently conducted the experiment, and they discovered that in order to complete the action of antibodies, or in order to complete the functioning of antibodies, it requires certain serum components. Okay, so they are heat sensitive serum components, so they named it as complement, since they are supporting or complementing the activity of antibodies in our serum. Okay, if you look into the functions of this complement protein or complement system, the major function is cell lysis. Before that, so whenever a complement system is activated, various components in the complement system, they start interacting each other, and that leads to a highly regulated cascade of events that helps to carry out this number of basic functions. First of all, cell lysis. That means whenever an infected cell or even a bacteria or a virus which enters our system, this complement, it is foreign to our body, right? So the complement will go and they will recognize us, and they will bind to this cells and causes the complement mediated lysis of the cells. And the second important function is opsonization. So you just look into this marked portion. So this indicate opsonization phenomena. Whenever a bacteria, you can see here in green color, which enters our body, the complement system will recognize us and binds to the surface of this bacteria. It's a kind of marking. So once this complement binds to this bacteria, these bacteria were marked now. So the receptors for this specific complement factors actually located over the surfaces of phagocytic cells. So this complement will bind to the phagocytic cells and enhances the phagocytosis and destruction of this complement marked bacterial cells. So this phenomenon is actually referred as opsonization. And this activation of our complement system very oftenly leads to the activation of inflammatory response, because that also helps to eliminate the antigen or pathogen from our body. So inflammation usually occurs due to the mast cell degranulation. Mast cells means a kind of immune cells. So this mast cells also carry receptor for the complement over their surfaces. So whenever a complement system activated, that leads to the release of various activated fragments of complement. That fragments will selectively go and binds to this mast cell complement receptor, activate the mast cells. Once the mast cells activated, it leads to the degranulation. Degranulations means mast cells were stored with various granules in them. Actually, these granules were filled with inflammatory molecules like histamine. So whenever this complement binds to the complement receptor over the mast cells, this granules will break, and that will release us inflammatory molecules like histamines into the surrounding environment. That leads to the inflammatory response. And even neutrophils also carry the receptor for complement protein. So whenever a complement system activated, this complement fragment or protein will binds to the receptor over the neutrophil surface and enhances their extravasation. Extravasation means their escape into the site of infection or site of injury by a process called diapedesis, or they slowly migrate between the space between two adjacent endothelial cells, because during this inflammation or inflammatory response the vascular permeability increases, so that these neutrophils can easily migrate through the space between the endothelial cells into the site of infection. And the fourth important function of this complement system is clearance of immune response or clearance of immune complexes, not immune response, immune complexes. Whenever an antigen, which I'll make you clear by some drawings. So whenever an antigen which enters our body, what happens? This antigen is recognized by initially specific antibody, right? So the antibody will mark this antigen. This antibody very oftenly carry a binding site for the complement components. So now the immune complex means antigen antibody complex, it is marked by the complement system or complement protein, right? So next the same procedure, since the phagocytic cells which carry the receptor for this complement protein, this complement will binds to the receptor and enhances the clearance of immune complexes from our body. Next let us look into the major components of the complement system. Mainly the complement system works by the help of a group of soluble proteins and glycoproteins. Mainly the soluble proteins, generally known as complement components, they are synthesized by liver hepatocytes, blood monocytes, tissue macrophages and by certain epithelial cells, especially epithelial cells of our gastrointestinal tract as well as gastrourinary tract. The serum components which constitute nearly 5% by weight of the total serum globulin fraction. Mainly this complement components which circulate in our serum in an inactive form, it is known as proenzymatic form, or they are called as proenzymes or zymogens. So whenever this complements were activated mainly by the proteolytic cleavage of this complement, it leads to the generation of one activatory subunit and it leads to the release of inhibitory component. That means during the time of proteolysis, the inhibitory component selectively removed from the complement protein, and that leads to the exposure of the active site of the major complement component, and that mainly participate in the complement cascade. So mainly the complement components, they are named as mainly named by using letters and numbers. So there are a range of complement factors or components. They ranges between C1 to C9, like complement components were named like C1, C2, C3 till C9 it include. So as I discussed before, these complements are mainly in their zymogenic form. During the time of activation, it cleaves into fragments. So one subunit is the activatory subunit. So once a complement component, zymogenic form of the complement component cleave, that leads to the generation of a bigger fragment and a smaller fragment. So bigger fragment always named B, small letter with small letter B, and the larger the smaller fragment which is marked with the small letter A. Mainly the smaller fragment which participate in inflammatory response, whereas the bigger fragment which is named B, which participate in the complement cascade by interacting with various other activated complement subunit. For example, whenever the C3 complement factor is activated, it leads to the generation of C3A and C3B. Okay, so B is the larger subunit which participate in complement cascade, and A is the smaller subunit which participate in inflammatory response. The case is very similar with C5, C4 and other complement factors also, but there is an exception in case of C2. Whenever the complement factor C2 is cleaved, it leads to the generation of C2A and C2B, but where the C2A, the A fragment is the larger fragment. But see, generally A fragment is the smaller fragment, but in case of C2, A fragment is the larger fragment, since it is the larger fragment which participate in complement cascade, and B is the smaller fragment which doesn't have that much important functions in our overall complement activation pathways. So this is all about our complement system. It's a kind of brief introduction about the complement system and the major complement components. So soon I will be uploading videos over the different kinds of complement activation pathways.